Okay, in Environmental 200 this week or the past couple weeks, we've been doing a bean lab experiment. I chose black beans to do this experiment. The two pollutants I chose to use were coffee grounds and sugar. With these two pollutants, I made like a concentration, kind of like what we did with the salt. I talk about why I chose to do these things, and sugar can mimic the overpowering of the nutrients in a day-to-day life that plants actually receive, and coffee grounds can be seen as organic waste in the soil, so I thought maybe that would be a good option to use. For the design of this project, I took 3 black beans per baggie. I did 2 sugar water concentrations and 2 coffee water concentrations, and then I did one of just regular tap water. Actually, yeah, I did tap water. And then for the sugar water solution, I did 100 milliliters paired with 6 grams of sugar, and then same with the coffee. I did 100 milliliters of water paired with 6 grams of the coffee grounds. For the 1% solution, I did 3 milliliters of the pollutant, 7 milliliters of water, and for the 4%, I did 5 milliliters of the pollutant and 5 of the water. I added the solutions to the beans, which were wrapped in a paper towel, so essentially I saturated the paper towel, and then I tracked the results. My hypothesis was that both the sugar water and coffee grounds would have a negative effect on the seed growth because sugar water might create the growth that harms the seeds, and coffee might lower the pH level of the soil. While I didn't track the pH level of the soil, it kind of got covered. These are my growth tables. You can pause to read, but as you're going through it, none of my things started germinating until the 28th, and that was only the pure water. Anything that had my pollutants in it did not do anything, except honestly crack. These are the summaries of my results. A lot of them did not have germination or growth. Essentially, like I mentioned, a lot of them cracked, and the only one that did have germination was the 100% water. This is my graph just showing I color coordinated it. The coffees are in browns, the sugars are in pinks, and the water is in blue. And as you can see, the water is the only thing that's going up in the growth rates, and I measured that in centimeters. Was my hypothesis confirmed or denied? With my hypothesis, I would know whether it was confirmed or denied by the growth rates, which I mentioned. So if there was stunned growth, my hypothesis would be confirmed, and if there were a significant amount of growth rates, my hypothesis would be denied. Altogether, my hypothesis was confirmed. The only growth rates were the 100% water, like I've said 100 times. Even that bag didn't grow as much as I thought it was going to grow. I mean, it had a little germination, but for some reason I was just thinking within a week a bean would grow more than it ended up growing, which is fine too. If I did this experiment again, I would just make sure I researched the best growing hacks, maybe what light it needs, what temperature it needs, solutions, etc., to see success, because with my radishes and now my beans, I haven't really seen much success, and it kind of saddens me because it's a lot more fun when you're having success, right? But the one thing I would say was maybe that I messed up was I saturated my towels, I think, a little too much. There was no extra water, but when I unfolded the towels to measure the seeds, it was really damp, so